Introduce Wut Phue, who is a PhD candidate in food science and agricultural chemistry, and she's going to be talking about the implication of silica and titania particles in food and their effect on human nutrition. And with that, the floor is yours.
Thank you, Andre. What is nanoparticles? You might think that the nanoparticles is a small, tiny powder, nanomedicines, quantum effects, material science, and safety, and so on. According to the ISO definitions, material with any external dimension in the nanoscales or having internal structures or the surface structures in the nanoscale called nanoparticles. If we are putting on the actual scales, nanoparticles are the 1,000 times smaller than the diameters of human hair. And when the size of material is reduced to the nano range, there is an increase in the surface to volume ratios, and the numbers of atoms on the surface give the novel function to the nanoparticles that are not usually exhibited in their bulk counterpart. Due to their novel functionalities, nanoparticles are used in the many applications such as energies, electronics, medical device, pharmaceuticals, and agriculture and food industries. Right. Is really nanoparticles are used in the food industries. And yes, in the food industries, nanoparticles are used in the farming stage up to the food packaging stage. And there are lots of nanoparticles present in the food products, and we are eating every day. But we may not realize this because they are not intentionally added as a nanoparticles, or they are not labeling as a nanoparticles. They usually add as the inorganic food additive materials. Even those inorganic food additive particles are officially approved. In those food additive, nano-size fragments particles are also involved. And according to the reference, around 40% of them are in the nano form. And studies of the adverse health effects of those tiny size particles are still ongoing, and especially the immunogenicity effects of those particles are overlooked. That's why today I'm going to present about the implications of silica and titania particles in food and their effects on human nutrition and health. When the nanoparticles are added to the food product, and it forms the protein corona onto the surface of the nanoparticles. And those protein corona nanoparticles includes in the food, and that goes into our bodies along with the food. So the main question is the corona protein will be the same nutritional value and the health effect as the normal proteins. And to answer the questions, I will study the adverse health effects of McGill titanium particles. And in this experiment, I didn't use the M&M chocolates or the lollipops or candy, which I presented before. And I have used the skim milk because this is more simple than the other food product. In terms of the adverse health effects, I studied the allergenicity effects of milk proteins because milk allergy is one of the most common allergies for kids. To know about the milk allergy in the presence of titanium particles, I studied the 4 steps, which are the characterizations of corona formations onto the titanium particles. And the second one is the antigenicity test. And the third one is the antigen structural conformation. And then finally, the fourth one is the allergy response of the cells. In the following slides, I will explain more detail of each steps. This is the visual image of the biocoronal formations of the titanium particles. And I also use the FDIR to quantify the biocoronal formations of the titanium particles. As we can see here, when the titanium is added to the mixed samples, the other biomolecule like proteins, lipids, and carbohydrates were attached onto the titanium particle and form the biocoronal onto the particles. So those biocoronal were used for further study like antigenicity and allergenicity tests. For the second test, the antigenicity effects of MYC proteins after interacting with the titania particles. I have used the enzyme-linked immunosorbent assay to study that, and this is the workflow schematics of the identifications of specific antigen response in the presence of titania particles. The reason I'm studying the antigen response is that antigen is the main component to cause the allergy, and most of the antigens are the proteins, and there are different antigen for the different allergy response. For the MYC allergy, beta-lactoglobulins and caseins are the main antigen for giving the MYC allergy response. The results show that the antigen response was increased in the presence of titania particle for both beta-lactoglobulin and casein compared to the control protein, which doesn't have the titanium particle, which I show as the dotted line, the levels of the antigen response without present in the titanium particles. Given the knowledge from my other studies and then the literature reports, this increasing the allergy antigen response may be due to the structural transformations of the antigens. For this reason, the structural transformations of antigens after interacting with the titanium particles were studied by using the FTIR and free thiol contents, which are representing the stabilities of the antigen structures. Based on those analyses, the conformational structures of antigens were transformed due to the interaction with the titanium particles. Finally, we have reached to the most exciting part of my study, which is the allergenicity studies. For this study, I have used the LED2 human mast cells and studied the release of enzymes, which called the beta-hexamidase enzymes from the cells, and which after exposing the antigen to the cells. This beta-hexamidase enzyme is the representative of the allergy response of the cells. As we can see, the allergy response was increased in the presence of titanium particles compared to the mixed samples. And I also did another test whether this allergy response was still the same even after passing through the in vitro digestion system, because the proteins or the other biomolecule were got eaten and got digested by the digested enzyme, which is present in our gastrointestinal tract. And due to that protein got digested, sometimes the allergy response were decreased. But in my case, the allergy response were still retained even after passing through the in vitro digestion systems. This may be the nanoparticles help the proteins got not digested by the enzymes, and that is the most interesting part of my studies. And as I mentioned before, I studied the two commonly used food additive dietary nanoparticle, which is the titania and silica particles. Just now, I was presenting only the results of the titanium particles, and the silica particles also show their similar effects. Okay, finally, the most complicated part of my presentation is done. But if you lose the track during my results presentations, don't worry, I will summarize the important point in the next slides. To conclude for this studies, when the antigen is interacting with the dietary nanoparticles, the antigen structures was transformed. Before I'm going to conclude the next steps, I would like to introduce the new terms called the epitopes. Epitopes are part of antigens that are mainly bind to the antigen receptor and giving the allergy response. The study suggests, my study suggests that due to the antigen structural transformations, the epitopes of the antigens were exposed to the antigen receptors. And when the more epitopes are accessible to the antigen receptors, it gives them more allergy response. And surprisingly, that effects they retain even after passing through the in vitro digestion systems. And the observations of my studies relates to the increasing rate of food allergies globally associated with the consumptions of processed food, often containing the inorganic nanoparticles. And that answer my research question, which I mentioned before. The coronal proteins will not have the same nutritional and health effects compared to the normal proteins. For this reason, we need to study more deeply about the use of inorganic nanoparticles in the food products. And thank you for your attention.